All right, hello everyone. We're going to be going over chapter 16 here. This is about how populations evolve. This is a little bit more in depth with how evolution works and the actual definition of evolution. So I know we spent a whole chapter going over natural selection, but this is where we're actually going to really dive into evolution. All right, so the first section actually kind of looks at Hardy-Weinberg. This is an equation that we use to calculate for evolution and if evolution is occurring. So basically, evolution is just any change in the allele frequency in a population over time. This is why we can say evolution is fact because we can visually observe this in a population. So again, we're not looking at major evolutionary trends. We're just looking at how this allele frequency changes again from one generation to the next. If there is a change, we can say that evolution has occurred. Gene pool, all of the alleles from the organisms in a population. I always find this is to be a funny looking diagram because like no one could understand this diagram without this kind of context of gene pool. So this is all the alleles. You can see here all these alleles are accounted for each one of these organisms. So you can see here in this generation, we start out with about half and half. And then you can see by generation 20, we're having a lot more of the recessive alleles here. So evolution has occurred in these populations. All right, this is where we're going to get a little bit mathematical, and I promise you it's not that bad. But in a population, you have the dominant versus a recessive allele. P is going to represent the dominant allele, while Q is going to represent the recessive allele. And our first equation is P plus Q equals 1. And just think about 1 as being 100%. When a population doesn't change, we call that the Hardy-Weinberg equilibrium, and we use this other equation. So basically, we have p squared, which is your the frequency of your homozygous dominant genotype, 2pq. This shows you the two heterozygous genotypes. We'll understand why there's two here in a second. And then with the q squared, this is just your homozygous recessive genotype. That equals 1 again. So we're going to use those two combinations or those two alleles in combination. Uh, you can see here, I'm sorry, those two equations in combination. You can see here we have the heterozygous versus heterozygous. We have the homozygous dominant, homozygous recessive, and the two heterozygous. That's why there's a 2pq here and only one q squared, and p squared. So you can see here again, p squared, q squared, then there's 2pq. That's again why we have the 2pq and not the 2p squared, if that makes sense. All right, so these Hardy-Weinberg equations. I'm not going to go through this right now, but basically, you have to use the Hardy-Weinberg equations to figure out when you get one type or one genotype, you have to figure out from there what that would be. All right, so the Hardy-Weinberg equilibrium. So this is going to be a kind of a condition that's met if no allele frequencies are occurring. So there's no mutations, so no mutations arise. No migration, there's no incoming or outgoing organisms that would change the gene pool. There's also a large gene pool. We don't see a small gene pool being enough to, like if there's a large gene pool, if there's not enough, let me go back. If there's a small gene pool, there's not enough organisms or there's not enough, let's say, variability where we're going to get this equilibrium. So in like small gene pools, certain things can happen. Like we're going to talk about genetic drift here in a little bit, and that just messes kind of everything up. So we need large gene pools. Random mating. This is just randomness. There's no like choice here. Everything's just random. And no selection. There's not artificial or natural selection going on. If all of these are met, Hardy-Weinberg equilibrium is occurring. But what you're going to see is it never really occurs because there's always evolution going on. So you can see here again, we're just going to go over it. No selection, no mutations. So you can see that third eye. There's no migration from one area to the next. There has to be a large gene pool, and there has to be just random mating. There's no choice in this matter. Yeah, so it really doesn't. Again, I love this meme somebody created. It said it really doesn't occur in nature. That this doesn't happen. All right, moving on to gene flow. This is where we're going to see movement of alleles between populations. This is that migration in or out of a new population. You can see here we're taking this allele or the alleles in this organism and we're going to the next population. Genetic drift. This is just any kind of genetic or random event where like there wasn't any purpose to it. So you can see here this person just kind of randomly stepped on the two green beetles and now we change the allele frequency. There is no it's not being better like is tan or tan's being better. It's just it happened that way. And I'll show you again an example. So like this boulder fell down and crushed all those trees. This changed the allele frequency. Now there wasn't any, you know, betterment for those trees to be there. It was just random where that rock fell and killed those trees. So again, this is just random events. Another type of genetic flow that we see is bottleneck effects. So whenever a population goes through a huge drastic decrease in the number of organisms, you just decimate the genetic variability here. And it's just due to chance. You can see the original population has a lot more genetic diversity than the new population. And it's because if you think about a bottle, you're only going to get a couple out of that bottle for the next generation. So it's just going to kill genetic diversity. Another example of gene flow is the founder effect, where you have a small population founding the next population. You can see here, this is a lot. There's a lot of genetic diversity here, but the new population is going to be kind of only a small portion of that, and you're not going to get much of that original population's diversity. All right, stabilize, or I'm sorry, the next section is it's chapters 16.3. So this is types of natural selection. In this section, we're going to go over there are a couple of different types of natural selection that occur, and they're how they look on a graph. So you can see here, we remember that bell curve that we've talked about before. We usually get this set of data here, and what happens is the outsides are selected against while the insides are selected for. This is called a stabilizing selection. And the example I give is human birth weight. So like it's not very advantageous to be very heavy when you're born or very light. The death rates there are very high compared to the death rates being an average baby. So you can see here the death rates being very large or very small are a lot higher because again there's a lot more complications in pregnancy, and this is not a good thing. This is, and we've actually gotten better with this. Our medical community has gotten really good at saving these babies, but just in general, being large or being very small, you have a higher chance of dying at that age. You have a lot better chance of surviving at these average age or these average weights. So there is a large portion of the average, not a large portion here from the outsides. The next is directional. This is when you can see here the shift is towards one direction or the other. So this section here in the red is selected against, the blue is selected for. So over time, we're going to see the direction of that move. And horses are a great example of this directional selection. You can see over time, the body size of horses has dramatically increased. And we have a lot of fossil record for this. So again, you're pushing the average to the right here and getting larger and larger. All right, our last one's disruptive. This is when the two outsides are actually selected for and the inside is selected again. So over time, you're gonna get these two peaks. So in this butterfly population, it's very advantageous to be very light or very dark, not in the middle. And you can see, I'm gonna show you here in this example, again, it's better to be very light or very dark, not in the middle, just because, again, whatever the environment is, it's better to have a dark fur here or a light fur, but not in the middle. Another type is sexual selection, and this is when females actually choose their mate, and this is when you get these kind of broad ornaments. In these species, the males will have these, you get just extravagant, like in this peacock here, these eyes here. We see the more eyes or the larger of this. I don't even know what you call it, plume. I forget what this is called, but the tail is a lot larger. They'll get more mates. So this is just again sexual selection, and it, it's actually kind of crazy that when evolution was just being hypothesized, that a lot of people didn't accept that females had this choice. Again, this was a very different time period. Males and female dynamics there that back then was a lot different, and basically what we learned is that was not obviously true, and females do have a selection force. We see this in I think it's called the Irish what is it called? The Irish deer, the Irish elk, where they had these humongous antlers, and it was because of sexual selection. It actually drove their species into extinction. But this again leads to males competing, and we talked about this with Erasmus Darr when he saw this as well. Which leads to again sexual dimorphism, where you're going to have not all species, but some species will have like male and female characteristics, which will help the females choose the best males in their eyes for copulating. Nope, I'm not watching that. All right, here we go. So one concept I like to go over with this section is that evolution doesn't produce perfect adaptations. There are always problems with these adaptations. So you can see like our backbone. We didn't evolve to stand upright from the first get or from the first species. We evolved to be kind of horizontal with the land. And now that we stand upright, we have these problems. So again, it's not perfect. You're going to have problems. And there's a really cool video where it shows this nerve in a giraffe that runs from the neck all the way down to the heart and back up. Where it's if it would just go like 2 inches, it would bypass that whole trial or that whole distance. But again, evolution isn't perfect. And again, this is just more evidence that supports evolution. All right, exaptation. This is exactly what we're talking about. This is how natural selection either takes a adaptation and it either improves it or it changes it. So the use of it changes. So originally the feather here was not always the feather we know today. There's a stage that we see in this evolution of the feather, and it was used for thermal regulation first, but now they use it for flying. So again, the original adaptation was not for flying. We changed it though because then it'll help the dinosaurs allow them to fly. All right. Lastly, here is heterozygous advantage. This is if the heterozygous of a genotype or a disease is actually advantageous to have. I know it's very difficult to understand this, but like you might think like, well, if these if evolution is true, like how can we have these deadly diseases? Like there would be no advantageous benefit for having this disease and it would be weeded out very quickly. Well, in certain parts of the world, certain diseases are actually advantageous to be heterozygous for. To give you an example, malaria and cystic fibrosis are two of these examples. Cystic fibrosis, if you remember, the lungs fill up with mucus, and it's because they don't allow these ions out, which there's a lot that goes into this. But basically, this helps people that have cystic fibrosis or have the heterozygous form of it be kind of immune towards what is it? It's cholera. Cholera. Same goes for malaria and sickle cell. If you are heterozygous for sickle cell, you are you have higher immunity against malaria in these populations. You can see malaria is a really there's a lot of malaria in Africa, and we see this sickle cell trait around these malarial places. Okay.